Hi, my name is Ruby Yu, and I'm a postdoc in the Marson lab at UCSF. Today, I want to tell you a story about you, or, well, your DNA. It's a very simple story, but it's actually so simple that it's complex, so I'm going to explain it using a few analogies to art and theater. Visualize a digital rendition of the Mona Lisa. From a distance, we see one coherent image, but now let's zoom in, and we realize that the image is actually composed of many individual pixels. Say we want to write some simple code telling a computer how to generate this image. Put a brown dot here, then a black dot, and so on. That would be straightforward. But now consider a more complicated scenario. Instead of one set of instructions for a single computer, imagine that the Mona Lisa is actually represented by many linked computers, each with its own set of instructions. Seems a little complicated, but still doable. The catch now is that every computer has the exact same set of instructions for how to become any color under the rainbow. So how does one computer know to show black and another know to show tan? Somehow, a network of blank computer screens, all containing the same code, must self-assemble into a representation of the Mona Lisa. Turn this two-dimensional image into the living, breathing woman that Mona Lisa was based on, and we have the puzzle underlying all human bodies. We're made up of trillions of cells, but they all share the same exact genetic code. So how does the same DNA tell an eye cell to become an eye cell and a heart cell to become a heart cell? To understand this, we need to get familiar with the different players in your genome, the most famous of which is the gene. You've all heard of genes before, and you know that they're associated with heredity, health, and disease. Your genome contains thousands of different genes, which can be on or off. What distinguishes an eye cell from a heart cell is the combination of genes that are on or off in each cell type. If you think of genes as actors in a play, then cell types are defined by who's on stage. But plays aren't static shots, they're dynamic. Actors come on and off stage at specific times in response to specific cues, sometimes alone, sometimes coordinated with other characters. They can't just wander on to any part of the stage, they have particular locations assigned. Like stage actors, genes are the stars of the show. But behind the scenes, there's a crew, unsung heroes ensuring that the actors show up at the right time and place, all according to the script. I'm interested in the script and the genetic stagehands that regulate when, where, and how genes turn on and off. Both fall under the umbrella term of noncoding DNA, and this is what allows your genome to code all the different cell types in your body and for all of them to function normally so that you are the picture of health. On the other hand, most of the times that something goes wrong with your body, leading to disease, it's because of an error in the script. Mutations in your non-coding DNA cause a system failure because genes are now wandering on stage when they're not supposed to, or missing the show completely. We know this, but because we still don't understand non-coding DNA, we currently can't do anything about it. The goal of my research is to decipher the code of non-coding DNA so that we can identify these errors in the script that cause disease and then use this information to create new therapies. The most exciting class of new therapies are cellular. As you know, your immune system keeps you healthy, defending your body from invading enemies, whether foreign, like a virus, or native, like tumors. Using recent advances in gene editing technology, we can now correct mutations or insert novel DNA into patients' blood cells, effectively hacking the immune system. doctors and researchers are now re-engineering immune cells to attack different types of cancers with incredible success. Currently, these interventions focus on correcting or inserting genes. Now, this is already game-changing, but modifying genes is only one approach, and quite a blunt one at that. You have a working gene or you don't. I envision a future where we can engineer not just the genes themselves, but also the non-coding DNA, the behind-the-scenes genetic script that dictates the nuances of when, where, and how genes enter the cellular spotlight. Being able to control genes to the same degree that nature does would allow us to not only tackle diseases where genes are broken, but also the huge class of diseases where the stagehands or the script are broken and genes are coming on in the wrong contexts. In order to bring this future closer to the present, I'm using a gene editing technology called CRISPR to intentionally mutate non-coding DNA and figure out which mutations are harmless and which cause a critically important immune gene to either not turn on or to turn on abnormally. This information will help us put together a Rosetta Stone for non-coding DNA so we can identify the words and grammar and genetic stage directions and eventually write our own scripts to combat disease.